Guys, welcome to Alveolata. Today we're going to talk about the three groups in Alveolata. There's more groups, but we're going to talk about three, but we're really just going to talk about dinoflagellates because I have been teasing you about dinoflagellates all year, so today is the big day. There is a video that I'm going to post online so that you can get a really good sense of why I'm so crazy about them. And here are the learning objectives today. I want you to know what the three major lineages are in Alveolata, why or how are the dinoflagellates unique, how does bioluminescence in dinoflagellates specifically work, and what is red tide. Okay, so we're just going to focus on one group today. This group is called alveola or alveolata because they have alveoli or kind of little pouches, vesicles on the periphery of the cell and the cell membrane. And in the ciliophora, in the ciliates, this gives rise to all the cilia. Think of paramecium, all fuzzy and covered with cilia. But in dinoflagellates, these alveoli become something very different. They become cellulose plates. They're called theca. Okay, so there is an example of a ciliophora, really cute and really fluffy. And here is a dinoflagellate. Let me move my face. I mean, look at that creature. There, I've got more pictures of you. Dinoflagellates are so beautiful. You can't even see how beautiful it is based on that picture. Okay, I'm just going to tell you a little bit. Apicomplexa is a group in the Alveolata. We're not going to talk about it today. We talked a little bit about it in the paper last week, but there's some really common human pathogens like malaria, cryptosporidiosis, and toxoplasmosis. These are protists in the Apicomplexa, but we're not going to talk anymore about them. Similarly, we're not going to really talk about ciliophora, even though these are really cool little organisms. Paramecium is one of them. Stentor, this guy down here, is a really cool organism. They've got fine hair like cilia all over their bodies, and they're mostly unicellular, and they're mostly heterotrophs. I mean, even though they're unicellular, some of them are pretty big, like Stentor is huge. And they typically have, if they're green, if they're photosynthetic, is because they have endosymbionts. They don't typically have their own plastids. That's all I want to talk about ciliophora because I really want to spend the day talking about, oh yeah, so here's an example of a paramecium, right? This looks like it might be a million plastids inside, but they're not. They're actually distinct organisms. So they're endosymbionts. Okay, let's spend the rest of the class talking about dinoflagellates. Dino is like terrible, right? Dinosaur is terrible lizard. Well, dinoflagellate is terrible whip. And they have these stiff theca that can be either armor-like or sail-like in Serratium. But I want to show you more pictures of them. That's Serratium. They're marine and aquatic, mostly marine. They're mostly flagellated, but they can be non-flagellate and they can be filamentous. Here's some examples of some flagellate. They have two flagella, and they have one whip-like one, which is obvious, and then they have one that's around the median. It's lateral in a groove, and they're crazy little swimmers. But here are some pictures of how crazy these organisms are. Look at these theca are so beautiful. They look like helmets. They look like spaceships. They look like ancient art. They're incredibly beautiful, and that's one of the reasons I love them so much. You know, on top of their bioluminescence, these are really incredibly beautiful little creatures. But they're not always so beautiful. Here's a really good kind of horrific example of Blistonium. It's a maricide of copepods. So, right, copepods are tiny little organisms, right? Teeny, teeny, tiny. I don't know, maybe this is like, you know, I don't know how big these copepods are. They're like maybe 100 microns or something like that, 200 microns. And they have this monster dinoflagellate parasite inside of it, and you can see it, right? Just taking up practically its whole body cavity. So they don't always have theca. They don't always have flagella. They have kind of a very confusing form of nutrition, right? So if you look at this original diagram from our class, it puts them down here, the Dinophyta, as tertiary endosymbionts. But they're not necessarily always tertiary. There's some idea that they might be secondary. There are some examples for sure that they're tertiary. But they can also just have endosymbionts too. So, and they can be pure heterotrophs. So they kind of do it all. They're a very strange group. So half of them, basically half of them are strict heterotrophs and they never photosynthesize themselves. The rest are mixotrophs, which means they can go between heterotrophy and phototrophy. One of the interesting things about them is they contain pyridinin, and this is a pigment that's only found in diatoms, which is one of the first clues as to how they became tertiary endosymbionts. They usurped a diatom. Now, some of them actually have living diatoms in them still. So there are examples where there was historical examples where they stole the plastids from diatoms, but then there are examples that still have diatoms as living organisms inside their bodies. Such strange little creatures. Okay, oh, I'm saying some stuff. Yeah, so we don't know for sure. There's definitely been multiple evolutionary events in these creatures, but we don't know for sure yet. And this is partially because we still don't know, you know, the extent of how many dinoflagellates are there. We don't know the full extent of this group of organisms. Okay, but we do know definitely there has been some tertiary endosymbionts, and they have been separate events, right? At the bottom here, we have an example that a secondary dinoflagellate ancestor engulfed a green alga and became tertiary. And there's other examples of them encompassing a cryptomonad, hepatophyte, and a diatom. Okay, so this is a very promiscuous group of organisms. They got their plastids a variety of ways, and that's a major message I want you to take home from this today. Right, I've already said this. Sometimes they have a diatom endosymbiont instead of plastids, right? And they actually have the organism in there, and it's just a regular endosymbiosis. And, you know, maybe it's intermediate on its way to becoming a plastid, or maybe it just works just fine as an endosymbiosis. Now, dinoflagellates are very strange. Their genetics are very strange. They have something that's called a dinokaryon, not a dikaryon, which you might be familiar with from fungi, but a dinokaryon, which is specific to the dinoflagellates. Most of the dinoflagellate body is the nucleus, which is interesting in itself. But what is more interesting is that the chromosomes are continuously condensed. They're always condensed. And they have no histones. They use a different protein acquired from algae to pack their chromosomes. They don't use histones. So just, they're very different than other creatures. And not only is their DNA different, these theca that I told you about, the theca plates, kind of think of them almost like, you know, continental plates on the planet, I think. They're these plates that are found in armored species, and where the growth of the organism occurs at the borders of the plates. So they add material to the borders of the plates, and that's how they grow. I just said that. Now, this is an example of, looks like paramecium, but dinoflagellates also produce some defensive projectiles, trichocysts. And these are little spears that are synthesized in their bodies and then ejected from the periphery of the cells. And they're beneath these, they're formed beneath the pores on armored species. So the theca have pores to release these trichocysts. But you think, how could an organism make such a defensive, sharp projectile and not destroy its internal structures? Well, they're developed in the Golgi, and they're stored in sacs which rupture on disturbance. So these Golgi protect them from the organism itself until they're attacked or there's some kind of damage, at which point they rupture and they're ejected through these pores. As soon as water gets in there, they grow really fast. You know those little sponges you can get at the dollar store that are like dinosaurs and things? You put them in the bathtub and they just expand. Well, that's what happens to these trichocysts, and they explosively release from the organism. And in the water, they become much longer and much thinner and much more harmful. So once they're in the Golgi inside the body before there's damage, they're really not that harmful to the organism. I mean, clearly it's some kind of defense. I don't know why papers in your text say they don't really know what the purpose is. Clearly it's defense. At least I think it's defense. Now, I just wanted to show you a little picture of the little cuties swimming around. I mean, there we go. They're champion swimmers, as I said. They can swim up to 500 micrometers a second. And when you think of how tiny these little guys are, I mean, this little guy's actually not a really good swimmer because he's just going in circles. Yeah, forget about that. Ignore that. Ignore that video, sorry. Okay, and there's a good example. You see the transverse flagellum and the longitudinal flagellum, and they're retained inside their grooves. Now, another neat thing about dinoflagellates, because you think about it, they're heterotrophs. A lot of them are heterotrophs, and they're stuck in these thecal plate-like systems. They have an extendable peduncle, and this is for attachment and also for feeding. So they kind of, you can see the peduncle sticking out. It's kind of like a snail or a siphon from a clam. They can exude this fleshy appendage for eating if it's a heterotroph. And there it is again. It's a phagopod. And here you can see 3 dinoflagellates eating this prey. They're all got their peduncles attached to this prey, and they're sucking it up. Okay, this is what I really wanted to talk about because I love this. I don't know if you've ever seen this, but I've seen it many times in my life, and I never get over how spectacular and how magical it is, bioluminescence. If you've ever been swimming, I've seen it in both the Pacific and the Atlantic. And at first you're not even aware of it until you start moving, and then suddenly it's like sparkles start appearing around your feet, and if you're in your whole body, there's ripples of sparkles as you're swimming. So spectacular, and it's all down to these little dinoflagellates. So this bioluminescence occurs in 30 photosynthetic dinoflagellates and some non-photosynthetic dinoflagellates. And I don't know why I have a picture of a copepod there. Let's just pretend it's not there. Oh, it's because it's a predator, sorry. It's thought to have evolved. What's the point of being bioluminescence other than it's this magical, wonderful event for humans? Well, it's thought that it reduces attack by predators by attracting their predators. So like a copepod, for example, this little guy that eats a bioluminescent dinoflagellate, it's also see-through, so it will start growing, and then the copepod's predators can see it. And so that just helps indirectly protect the dinoflagellates, because the copepods who eat these bioluminescent organisms figured this out, and they'll release the bioluminescent ones. So some organisms can avoid this, and they can still eat bioluminescent organisms because they develop this pigmented internal organs, which you think is kind of strange. Why would you need pigment inside? Well, it's so that you can trap the light inside your body and not attract predators. So some fish actually have black stomachs, so they can eat things like bioluminescent squid and cuttlefish and dinoflagellates. Okay, how does this happen? Well, it happens as a chemical reaction in the cell of the dinoflagellate. There's organs at the periphery of the cell in this cytoplasm. They're called scintillons. And inside the scintillons, the dinoflagellate produces a protein called luciferase, an enzyme, which when it's oxygenated, releases energy in the form of light. It's not that complicated. I mean, it is, if you think about it, how this evolved and why this evolved. It's extremely complicated, but it's really it's one enzyme, and it's one enzyme interacting with oxygen, and it produces light. And that's all I really want you to know about this. Here's a picture. I thought this was a good picture because it gives you an idea of what these organisms look like in the water. They're not the ones with theca, at least this one isn't. And they have these scintillons that are distributed throughout the whole body. And they call it a sea sparkle here. A single dinoflagellate can have up to 200,000 scintillons in the cell. Now this one particular has actually endosymbiotic algae in it too, which are not responsible for the bioluminescence at all. Okay, so watch the video. I'm gonna put a little video online just so you can see it and appreciate it more. You don't really have to go into much more detail than that. But another thing dinoflagellates do is they are responsible for red tide. Now, red tide, I don't know, when I lived on the East Coast, it was a big deal, right? There are certain times of the year you couldn't eat the shellfish because they were toxic. And the reason they were toxic was because of dinoflagellates. There's too many, these organisms are eating dinoflagellates, and the dinoflagellates produce toxins. So red tide happens when the concentration of these particular dinoflagellate gets so high that these organisms' flesh becomes toxic themselves. Why do the dinoflagellates get this high? Well, you know, fluctuations in their populations is totally normal, and occasionally algal blooms will happen in natural ecosystems, but they don't typically happen as much as they're happening now. I mean, they happen in nature because these organisms are really good at moving around, and they're good at phototaxis and chemotaxis. They can find the nutrients, they can find the sunlight, and they'll all migrate to the same areas when conditions are good. And when they're in a big group together, they're more protected because there's fewer predators. But these big aggregations of dinoflagellates are dictated by environmental conditions. And well, let me get to that in a second. Okay, so why are they toxic? Well, they produce a toxin called saxitoxin, and it's a neurotoxin, and it paralyzes shellfish in particular. And it makes sense because shellfish are eating these guys. So of course they're gonna have defense compounds. Unfortunately, they're also toxic for us and for other organisms up the food chain. And it produces an illness in humans called paralytic shellfish poisoning, which is why you can't eat these fish or these shellfish during red tide season. But it's important to know that red tide is natural and it's seasonal, and it happens in natural ecosystems, but it lasted 17 months in Florida recently. I mean, they're getting bigger, they're getting longer, and they're getting more frequent. I mean, look at this whale shark, this poor little whale shark. It's not just killing shellfish and making humans wait for their clam chowder, but it's killing dugongs, it's killing all sorts of creatures. And not only that, not only is it toxic to eat, but these dinoflagellates release toxins, aerosolized toxins into the atmosphere that are really damaging to lungs. And so, you know, some beaches in Florida and places in the South, they're wearing elaborate masks. Okay, so they're natural, but like I said, they're more frequent, they're bigger, and they're longer. And since the 1950s, there has been so many more of them, and it's largely due to the eutrophication of our oceans, right? We're pumping way too many nutrients into them from agricultural runoff, and our oceans are warming up. And these are creating really eutrophic conditions for dinoflagellates. I have some good slides, but they're at the end. Okay, now, who else is affected by these new ocean conditions, right? These hot, kind of very eutrophic nutrient conditions. Well, dinoflagellates have endosymbionts, right? We've talked about how they have endosymbionts inside their bodies, but they can also be endosymbionts. I mean, these guys are they're so flexible. And clearly I'm talking about coral. This is a picture of two coral, right? Coral are cnidarians, right? And we only think of coral when we think of those beautiful structures, you know, those calcium carbonate structures in the ocean. But if you want to look at the actual little cnidarian, there's these little tentacles, right? These little tentacles, and there's the mouth and these little tentacles. And the dinoflagellates live in the tentacles, and specifically in the tip of the tentacles. And what they do is they perform photosynthesis for the corals. Corals can't do that themselves. They're cnidarians. They're related to snails, slugs, octopus, things like that. Diatoms in coral are called zooxanthellae, and they perform the photosynthesis that make the coral reefs such spectacularly productive and diverse ecosystems. A single coral reef can contain up to 30,000 dinoflagellates per millimeter cubed. So coral reefs don't just associate with dinoflagellates. Coral reefs are packed with dinoflagellates. They are almost, you know, I don't know what the biomass ratio is, but there's a lot of dinoflagellates in coral. And dinoflagellates are responsible for the spectacular colors of coral reef. It's not the cnidarians. The cnidarians are kind of see-through, but the dinoflagellates have spectacular pigmentation. And really, it's the dinoflagellates that make these systems so unique and so wonderful. So obviously I'm going to coral bleaching. We're going to talk about it next week. This happens when the cnidarians, the coral polyps, expel their algal or their protestin partners, right? So in this case, I say algal, but I mean dinoflagellates. So they get rid of their dinoflagellates and they can't make any food. The whole food web falls apart, everything dies, and all that's left is the calcium carbonate skeleton, right? So here's a picture of a coral reef. This one's still alive, right? You can see all the little polyps. Inside those polyps, they're packed with dinoflagellates. When conditions get too hard, the polyps can't support them, right? Because the cnidarians have to feed the dinoflagellates. I mean, not feed them carbon, but feed them nutrients, protect them. It's too much work for the coral to do this when conditions are tough, and they expel them. So here's a little cartoon of it. So a healthy coral, the dinoflagellates are in the polyps. They exchange nutrients. It's a symbiosis. They're happy. Everyone's happy. The whole food web is happy. The whole ocean is happy. As soon as coral gets stressed, like by temperatures being too high, they can't regulate thermoregulation. There's toxins in the water. They start expelling their dinoflagellates because they can't afford to keep them, right? You have to pay your symbionts. They can't afford to keep them anymore. And ultimately, if conditions continue to deteriorate, there's nothing left. Once they get rid of the dinoflagellates, the coral cannot make food for themselves. They are heterotrophs, they're not autotrophs, and they can't forage for themselves. So that's how bleaching occurs. And this is what it looks like. I almost don't want to look at it. It's not good, guys. I already explained that. So when coral are very stressed, they expel way too many dinoflagellates and they die, and this is bleaching. Okay, so this is why it's not good. So here is a graph. You guys are probably familiar with this. This is atmospheric carbon dioxide, right? Until the 1960s, it was pretty steady. And then boom, with industrialization and fossil fuel use and human population growth, it goes up and up and up and up and up and up every year. I mean, it goes down during the winter, but up and up and up every year. And these lines are bleaching events. So the first one was in the 1980s. And these are mass bleaching, global bleaching events. But then 1998, 2010, 2015, 2016, 2017, they're happening every year now. And I don't know, I mean, I hope that you guys have seen the coral reefs because your kids aren't going to. And that sounds dramatic, but it's not. Here's a simulation of climate change. The red line is like business as usual, nothing changes, right? Which is where we are now, because even Canada is not meeting its commitments to the Paris Climate Change Agreement. So it's business as usual as far as I can tell. Here, the blue line is if we take some measures and reduce if we take strict, like the strictest measures that are possible for humans to take. Right? So these are both, this is business as usual, and this is if we totally get rid of fossil fuels and change our lives. If in the red version, business as usual, we lose corals in the next 30 years. They're gone from the planet. Version 2, we lose most of the corals, but some of them recover by 2100 to the change of the century. It's not good. It's not good. Sorry to leave it on such a downer note, but it's really important, I think, that we understand how permanent these changes are and how irreversible climate change is, so that we can do what we can. If we can't save the coral, maybe we can do something else. Anyways, I'm gonna go cry now, and I will talk to you later.